Sorghum is a C4 cereal crop. It is extremely drought tolerant. It is also flood tolerant. And it doesn't use the amount of fertilizers that other cereal crops do to produce the same amount of biomass. So it has a lot going for it in terms of handling climate change. I'm Peggy Lemaux. I'm a professor of cooperative extension in the Department of Plant and Microbial at UC Berkeley. And I work on a lot of cereal crops, but more recently on sorghum bicolor. One of the really important things about sorghum is that JGI has sequenced a lot of sorghum varieties. And what you can see behind me is a lot of different. So there's some like really short ones, some really tall ones, some sort of in between, and they all have characteristics that make them special. And we now have technologies to be able to engineer or edit all the sorghum varieties that we've tried so far. So it really opens up a lot of new places we can look for information. So when we first started working on sorghum, there was no genome sequence. And we were working on a Gates Foundation project where we were trying to improve the digestibility of sorghum. We knew there were proteins or seed storage proteins, but we didn't know how they were regulated really. We didn't know how they were related to each other. And so when the genome became available, that really opened up a whole new way of thinking about how you approach drought or how you approach a nutritional problem, because now you have a dictionary. You can go in and find out what the various genes, what genes are there, and potentially begin to figure out what they do and how they relate to each other. So that was extremely important. EPICON stands for Epigenetic Control of Drought Tolerance in Sorghum. And it started because sorghum is extremely drought tolerant, and we wanted to understand how it is that crop versus, let's say, maize, which is a close relative, how is it that sorghum is so drought tolerant? And so that was the focus of the project. And we also studied the microbiome. So we also wanted to know how microbes might also be involved in drought tolerance in sorghum. So we found a lot out about the plant itself. We had control growth, where we gave it all the water it wanted, and then we had drought conditions. And by doing that over 3 years in the field, we've been able to identify about 200 genes that are involved in either being upregulated or downregulated during drought. And so now one of our goals is to find out exactly what those genes do. In addition, we have a lot of information on how plants and the microbes in the soil interact and talk to each other. And that was something that we didn't know before. The amount of data that we had was immense. And if it hadn't been for JGI, we would never have been able to get the data and then to handle all of it as well. So the EPICON project is probably one of the most productive projects that I've ever been involved in.